All right, so I'm sure you guys all recognize me. I'm the guy who sits at the back of the room and doesn't really say much. My name's Tyler Kostiuk. I'm a master's student here in University of Alberta. I'm in the Faculty of Laboratory Medicine and Pathology. I'd like to apologize right off the hop because I know I do this a lot when I talk, and I always get yelled at by my supervisor and my co-supervisor, but I'm very energetic, very enthusiastic. All right. So I'll give you guys a quick rundown of what we're going to talk about. I'm going to do a little bit of overview, talk about what a drug is, what we're doing right now in terms of drug delivery, and then we'll move on to a couple of methods that are in development right now. The three that we're going to talk about are engineered viral vectors, a couple of other nanomaterials, and then implantable drugs. Because of the amount of time we have, I was going to ask if you wanted to take a break after each section to ask questions, because there are three completely different sections within the talk, or if you want to save it all till the end.
I think it's entirely up to you.
What do you guys think? Any ideas? Okay. We'll take a break after each section then. Give you guys a little bit of an opportunity to hop in. All right, so what is a drug? I actually got yelled at for this definition. My girlfriend's actually a pharmacist, and her definition is a little bit different than mine, but I'm not a pharmacist, so if there's any pharmacists in the room, I'm sorry, but this is what I have to work with. It's any chemical substance that can be used for treatment, prevention, or diagnosis of a disease, or to otherwise enhance the user's physical or mental well-being. I'm sure you guys could think off the top of your head of any number of methods of drug delivery. Time release in terms of capsules, so when you pop that Advil, Tylenol, depending on what your flavor is. Injections, topical and transdermal routes, and aerosols. I didn't really go too much into the history of it because I'm sure most of you guys know about taking pills and having flu shots and stuff like that, so kind of skipped ahead a little bit. That's where we are and where we were for the past 50, 100 years. But where are we going right now? In the past few years, there's been a major revolution in terms of nanotechnology. I did my undergrad at the University of Waterloo, and they actually just built a brand new nanotech building, and I think the budget for it was $100 million or something like that. But if anybody knows anything about Waterloo, they get all their money from RIM and BlackBerry, which isn't as much anymore, but it used to be a lot. There's been a variety of different approaches in terms of using nanotech towards drug delivery, engineering biological entities, typically bacteria and viruses. I've heard a little bit about using funguses too, fungi, but not as much. Engineered nanoparticles and transdermal patches kind of go hand in hand, as well as gene therapy. So the first one that we're going to talk about hits close to home with me because I work with one of the viruses that I'm going to tell you guys about, the majority of this portion of the presentation actually. There's two main uses. Most people know about the first one, using viruses as a means of delivery of gene therapy, so viral vectors. In this approach, you typically have a disease, a complex or simple genetic disease that arises either from the accumulation of mutations or from truncations to a vital gene. In viral therapy, you use a highly infectious virus. The main one they've been using lately is adenovirus, which has a number of drawbacks that I'll get to in a little bit. The idea here is you clone in a fully functional copy of the gene, you introduce the virus to healthy cells or unhealthy cells depending on the condition, and by homologous recombination you actually get the corrected gene inserted into the human genome, which is propagated down the cell line. The second approach, which will be what I'm actually going to focus on here, is using viruses themselves as the therapeutic agent. So it's technically still drug delivery because they're delivering themselves, but it's more about the actual use of the virus as a drug. In this case, we're using less infectious viruses. The example here is reovirus, which we're going to talk about right now. This is the virus that I work with in the lab. I don't do this kind of research. Again, I'm not a pharmacist. I'm not from the biochem side of things. But to give you guys a bit of background about the virus, it's a double-stranded RNA virus in the family Reoviridae. It's broken up into three serotypes, type 1 Lang, type 2 Jones, and type 3 Deering. They're not known to cause any diseases in humans. This virus has been studied for about 50 years now, and the initial thoughts were that it was a GI tract virus that caused diarrhea and any other general symptoms that you can think of. But in recent years, they've actually found that those were caused by symbiotic infections, so other things that were infecting at the same time, and reovirus typically doesn't have any symptoms in carriers, but it may be implicated in biliary atresia in newborns. That's a condition where you lack or have a deformed bile duct that connects your liver to your small intestine. Another important thing here is that it's frequently detected in both wastewater and human samples. Fecal, urine, cheek swabs, anything you could think of, you can usually detect the virus. This is important because it means the virus is already pretty much everywhere, which I don't really think is scary. When I told my parents about it, who have backgrounds in accounting, it kind of freaked them out a little bit, but again, different fields. It's detected by cytopathic effect in PCR, which is what I do. Now, to tell you guys a little bit about using it as a drug, if I can get the mouse out here. In healthy cells, when they're exposed to reovirus, they get engulfed and brought into the cells where they form the secondary particle, the infectious subviral particle. This outer capsid membrane then gets broken down, and its double-stranded RNA is released into the cells. In healthy cells, you have this protein here, PKR, which phosphorylates a second protein, E1F2 alpha, which in turn binds to double-stranded RNA and signals it for degradation, removal from the cell. So in healthy cells, you still get infection, but it doesn't go past releasing its RNA. It doesn't get translated, it doesn't get replicated. However, in certain cancers that have an activated RAS pathway, which is about anywhere from 20 to 45% of cancers, depending on what papers you're looking at, what happens is this PKR protein is deactivated by the activated RAS pathway, which means you do not get a phosphorylated E1F2 alpha, and you get translation of the viral genome, leading to replication and eventual lysis of the cell. And when you get lysis of the cell, it spreads to nearby cells, infecting everything. But what was important in there was that it has this innate ability, without being altered in any way, to actually preferentially infect and destroy transformed cells. So I don't know about you guys, but I came from a cell bio and biotech background, and to me this is pretty interesting to think that you have a virus here that naturally on its own, without any help, can infect and destroy cancer cells. I got pretty excited the first time I learned it, because I was already working with the virus, but another good thing about this virus is that it can be used in combination with other chemotherapeutics. One of the big ones right now is gemcitabine. These drugs work actually similar to reovirus in terms of infection. They have a similar active site. Gemcitabine is actually, it works at the microtubule level in cells. I don't really want to get into cell bio because I know everybody. There's a couple of people here that come from different backgrounds. But the long story short is they work in similar ways on the cells, similar areas anyway. So it's actually been shown to have synergistic effects when you use both of them intravenously together at the same time. A big benefit there is that if you have two drugs that are working together, you can decrease the dose of one that's potentially harmful, chemotherapeutic. They usually do a lot of damage to healthy cells as well as cancer cells. So the good, infection by reovirus suppresses tumor formation. Targets only cells with activated RAS pathways, which is 20 to 45% of cancers. You can combine it with other cancer drugs, leading to remission in 80% of samples. This was for melanoma. It also works for head and neck carcinomas and a variety of other kinds of skin cancers as well. And it causes little to no immune response in adults, and you'll see why that's important in a minute. The bad public perception, one of the papers that I used for my research paper was actually called Using Viruses Against Cancer, Turning One Scourge Against Another. So I'm pretty sure if you went in and tried to ask your doctor about what drug you're getting and they tell you, oh, well, we're just going to pump you full of virus, it might be a little apprehensive about that, but reovirus itself is prone to mutation and genetic reassortment. This is more important if you were talking about later on genetically modifying it to translate other proteins. And the ugly. In the late 90s, early 2000s, there's an 18-year-old patient, Jesse Gelsinger, who had ornithine transcarbamylase deficiency. It's a uric cycle disorder. Results in not being able to remove large amounts of ammonia. And he was subjected to adenoviral therapy. Adenovirus is a very infectious virus. It leads to still thousands of deaths around the world in the present time. 4 days after receiving treatment, he died of an apparent multiple organ failure brought on by massive immune response. So again, this is a different type of therapy, but it's still viral therapy. So if you tell a patient you're going to receive viral therapy, the first question they're going to ask, is it safe? Well, yes, no. And where are we now? I found it interesting that the properties behind this virus were actually discovered in the 1960s. There's two groups that were working on it. One of the groups was actually looking for a way to increase the effectiveness of the polio vaccine, which is kind of interesting. And nowadays, actually, at the end of stage 2 FDA trials, we have a drug called Reolysin from Oncolytics Biotech in Calgary. This is just wild type 3 reovirus Deering. And that's the end of the first section. If you guys have any questions about viral vectors, viral therapy, microphone.
Hey, I've always found it interesting when you're using viruses to try to treat something like cancer or something foreign, treating something that comes from your own body. I've noticed one of the problems with viral therapy is getting it past the immune system, and you were saying it wasn't very immunogenic, but also you were saying that the reovirus can mutate quite a bit. Is there any worries that this virus can be mutated to a point where it can infect normal cells?
Sure.
And if so, would they be able to say cure it if it was causing problems like that in treatment?
Yeah, well, the safeguard in your cell is that E1F2 alpha protein that binds double-stranded RNA. So unless they could find a way to circumvent that, that's the only way that I can think of right now to infect normal, or to cause damage to normal cells.
Yeah.
What was the first part of your question, sorry?
Well, you were saying that, I just wanted to confirm, you were saying it wasn't very immunogenic. So it didn't really...
oh, about the genetic variation, okay. I've looked at sequences that they've pulled in the past 15 years, and this is all, there's a high amount of variation everywhere in the genome. I do qPCR, so I have to do a lot of work with genomics and stuff like that. And I've never seen large-scale mutation that causes functional differences.
Right.
I'm sure it's possible. Anything's possible with viruses. That's one thing that I've learned since I've been here, unfortunately. But as far as I know, it hasn't been observed yet. Whether or not that'll happen.
Okay, thank you.
Sure.
So other questions?
Oh, one there.
So you mentioned that it only works when this ROS pathway is activated. Does that mean it's limited only to those kind of cancers then that have that? Or is there maybe a way where you can induce that pathway in cancerous cells to activate no matter what kind of cancer it is? Or?
Good question. Right now, it's only available for cells with an activated RAS pathway. Again, it comes back to the proteins that are affected. There's that, here I'll go back to the diagram. This PKR protein is the linchpin here. If you could find a way to deactivate that all the time, you'd have something there, but the problem with that is you're trying to target it only to cancer cells. So you have to get into cancer cells and deactivate the pathway only in cancer cells, which is, if you could do that, write your own paychecks for the rest of your life pretty much. But yeah, right now it's limited only to that.
Thanks.
My question is about antibodies. If it is that, if we have already infected with this virus, we have antibodies for this virus in our body.
Sure.
While you just put this virus inside, it will be just disarmed with antibodies. What did you do about that?
Sure. There's actually an interesting paper that I read a while ago about detecting antibodies in children and adults as well, but in children they actually have 100% of, up to 100%, it was hovering around 80 to 100% of newborns actually had antibody against IgG, I believe, they were measuring against reovirus. But by the time they reach age 5, 0% had it because they all lose maternal antibody by the time they reach age 5. I don't have a background in immunology. The girl in my lab does, so I ask her all the time. Anytime
You have memory cells over there then?
Yeah, we have, sure, yeah. From what I read in the papers, it's a really high dose because it doesn't cause a massive immune response, doesn't cause a large immune response. So they just increase the dose, that's as far as I know. I'd have to read into it a little bit more. I didn't dive into any of the immunology behind it.
Because this is the problem with the adenovirus.
Yeah.
If they are using adenovirus,
Yeah.
it will be just, they should think about
It's
it.
highly infectious adenovirus, and if you don't remove the virulence genes
Yeah.
in adenovirus, it replicates extremely fast in humans. So it causes a big immune response, not just because of the small dose, but because it's replicating so fast.
Other question that I have is that does this virus have any preferred site for infection in the body,
In the body?
any organ?
The GI tract, yeah.
Okay.
Typically, the big place, big site of infection is the liver, was from what I read,
Like
the GI tract.
adenovirus. Then you should think if there is any virus in other places, how to direct the virus to the other sites, not to the GI tracts.
Sure, yeah.
That's a problem as well.
Yeah.
Questions?
Oh,
So why don't you go on to the next then?
all right. The second part of the talk is about microneedles and microneedle arrays. A microneedle is exactly what it sounds like. It's a needle made on a micro scale. In terms of length, they range anywhere from the typical range here, but I've seen them all the way down to 50 micrometers, all the way up to, 1,000. They can be made of a variety of materials, steel, same as hypodermics, biopolymers, which are biodegradable, and silicon. It all depends on the application. The patches themselves are actually about the size of your fingertip. I have a couple pictures coming up, but they can house anywhere from 50 or so all the way up to thousands of microneedles. They differ from traditional needles in a couple of obvious ways. Depth of penetration is the big one. I have a diagram coming up showing that as well as number of points of penetration. They've been used successfully in a couple of different applications. The main one that I've been reading about is vaccinations. This has to do with the lack of specialized labor that's required to administer them, as well as transportation concerns. This is a biopolymer one. Just showing the size of it here. It has 36 points on it here. The cool thing about the biodegradable ones is once you have them on your skin as a patch, the needles actually dissolve. So after a period of time, they can be taken off and you don't need to use a sharps container. They can just be put with the rest of the biohazard waste. These are steel compared to on the right there, that's a hypodermic and hollow steel microneedles. And then lastly, these are the silicon ones that are actually etched out of silicon by a laser. And I don't know if you guys can see back there, but there's thousands of points on there. In terms of depth, your skin, outer part of your skin has three main layers. The stratum corneum, which is the mainly dead layer of skin. The epidermis, viable epidermis, and the dermis. Deep within your skin, this is where you start to, well, relatively deep, is where you get pain receptors starting to form. The dermis here, the bottom layer is around 150 to 200 micrometers below the surface of the skin. So the main issue here when you're talking about microneedles is breaking through the stratum corneum, which is 10 to 15 micrometers, but not going so deep as you break into the dermis where you're reaching the pain neurons and causing pain, physical pain. You can see here just the difference in depth too between the typical hypodermic needle and these new microneedle patches. This one's actually kind of interesting because you can see at the bottom there that it's actually sourced by HP. They're doing research into silicon microneedles. The upside is that they're less traumatic in a couple of different ways. If you're not reaching that level where you can start to feel pain, you're not going to feel pain, so there's nothing to be afraid of in terms of a physical feeling. The other side to that too is you have less emotional trauma, because I'm sure you've all, when you were little, you didn't want to go to the doctor to get your flu shot or go get any other immunization, right? So if you have a little kid and you bring him in and you show him a little patch and you put it on your skin, it's a lot less worrisome. Like I said before, you avoid a lot of technical costs because you don't need specialized labor. You don't need a nurse or a doctor or a pharmacist like they're doing now to come in and give everybody an immunization. You could just take a patch out of a wrapper, push it on your skin. Sharp disposal with the biodegradable ones. Again, just decreasing costs there. In terms of vaccinations, I didn't really get a lot of data about using them for delivering insulin or the cancer drugs, but in terms of vaccinations and T-cell response, they actually equal or exceed results given by traditional hypodermic needles. And then this is the big point here in terms of transportation and refrigeration, is that they can actually last out of a fridge for up to 6 weeks at a time. So if you're talking about bringing vaccinations to developing countries, to rural areas, you don't exactly always have the capability of pulling a beer fridge behind you and having a whole rack of vaccinations in there. This way you could take a backpack, you could take a bag, you could throw them on a bicycle and you could ride down the street and you don't have to worry about them thawing out. And this is very important when you're talking, same thing about if you're trying to immunize children, you're trying to eradicate certain diseases, you have to have technology that can get to them, which is why it goes in the big box and gets underlined. Limitations, again, people are resistant to change. This is nothing new. I know if I went in and asked for a flu shot and they started putting the Band-Aid on first, I'd be pretty confused, because that's what they look like in the majority of cases, is just a Band-Aid. Accountability, this is more of a logistics thing. I don't think you would ever come to the point where you could go to the pharmacy, get a flu shot, go home, put it on yourself while you're watching TV. I don't think it'll ever come to that point, but it's something to think about anyway. They're actually really fragile. In a lot of the experiments using steel and silicon, they've actually broken off points when they remove the patch, which again is why the biodegradable ones are really the way to go, in my opinion anyway. Scope of use. The one that I read about, the big one, was diazepam that they're looking at right now, trying to make it intranasal aerosol for epileptic patients. And you can't exactly go in with an aerosol, say if you need a nose spray for something else, you're not going to go put on a patch. So it's not an end-all be-all for all drug delivery methods, but it has a wide array of applications where it's very useful. Where are we now? The measles vaccine was going to be in the next 5 years or so. That's their goal for the first large-scale application. Like I was saying earlier, getting to those developing countries. In countries like Canada, the US, it's not going to replace the traditional MMR vaccines because there's no need to. We have the specialized personnel, we have the disposal units, might as well use them while they're here. In terms of cost of building all these new materials is going down. Research interest is going up as cost goes down. And we talk a lot in this course about unintended consequences. I'd like to be in the room when they were first brainstorming about making carbon nanotubes to see if they thought to themselves, I bet you we can eradicate measles when we make these. Probably not, but you never know. And that's it for that part of the talk. Any questions about microneedles?
Are there any plans for using it to eradicate polio?
Polio?
Yeah.
Not as far as I know right now. I didn't read any papers into it. Measles was the big hot button one for me when I was reading through all my literature. So I could look it up for you. Off the top of my head, I didn't come across anything about it.
It would just make sense because right now where it's endemic is in developing countries and
Sure.
places where people are really poor, and this seems like it might work for that last step.
Yeah, definitely. The refrigeration thing is key for developing countries. But like I said, I didn't stumble across anything, so I can't really give an educated answer. It would make sense, but...
So the kind of target area is in between those two layers of skin, yeah? Is that something that's variable
by person?
Between person?
Yeah.
Yeah, so what do they
go
do to combat that or to make sure that it actually works on everyone?
Yeah, that's one thing I forgot to mention. It is another limitation of it. Obviously, people with fattier tissues, I don't want to offend anybody, you know what I mean? They have a lot more of a buffer zone, right? Because there's that extra layer of tissue underneath. It's just a logistics thing. So it depends. You can't exactly go around the world and take everybody and cut off a chunk of their skin, right? And see, oh, what length of, it's not like fitting a shoe, right? But in a way it kind of is, because you don't want to go too deep, you don't want to go. But at the same point in time, the idea here is that the needles are fine enough, the tips, because you could see when I compared them to the hypodermics, that even if they do get into that bottom layer, it shouldn't cause that much pain. From what I read, people who did experience a sensation, apart from the adhesive on their skin, was that it was itchy. That was all I read about it. Nobody talked about being here. But that's a good point. I forgot to bring that up to you guys.
Thanks.
Yep.
Okay, so other questions? All right. So is that...
There's one more part.
One more part?
Yeah. Smart drugs.
Okay.
What am I doing for time? Okay. Is everybody having fun too? All right, so the last part of my talk here is about implantable medicine. It's a little bit more touch and go because there hasn't been that much research into it. The ideas for all this stuff kind of stems from the new technology, the labs on a chip that you hear about. I don't know if anybody in here does PCR. I do PCR on a daily basis. Yeah. Anyway, the machine I use is relatively, it's the size of a case of beer on a counter. That's the size of a PCR machine that we're using right now. And they've actually downscaled that technology to fit onto a glass microscope slide, where they vary temperature according to the cycle in PCR that you're in. So we're experiencing, again, a downsizing of technologies. We went from machines that were the size of a room to the size of your desk, to the size of part of your desk, now to it can fit together with other pieces. They're doing this as well for microarrays, which is a very powerful diagnostic tool in terms of genetics, cancers. So we're increasing the diagnostic power and drastically smaller spaces. The other aspect here is that if you're making something that was small and capable of doing a variety of diagnostics, you should be able to make it connect to other devices as well. So the idea here is you have an implant in your body that's capable of running diagnostics for a variety of conditions, monitoring different levels in your body, and you can connect via this implant to your computer, to your cell phone, to the doctor's office, to the pharmacy. And the last part here is the end goal is to build a unit that is capable of synthesizing and administrating drugs directly from within your body, which to me is a pretty tall order, because how can you fit that many components, that many ingredients into a tiny package? But now we're moving towards a pharmacy on a chip. So we want to take these implantable diagnostics and put them together with drugs inside your body that are ready to be delivered. The big example that I read about was streptococcus. So right now, if you go in to the doctor and you have a sore throat, they could take a swab, it's called a dipstick test, and ELISA. It's an immunoabsorbent assay. And from there, they can determine right away strep throat, not strep throat. So why not be able to take technology that's similar to this, put it in your body, Don't even have to go to the doctor. Boom, strep throat, okay, antibiotic. No need to go wait in line for the doctor. No need to go through a bunch of tedious tests, right away, move towards treatment. Limitations on this, there's a lot of them. I see a lot of flaws, and I'm not even working in this area. Accountability to me is the gigantic one. If you're sitting at home and all of a sudden your machine beeps and it decides to provide you a treatment and something goes wrong, who's accountable? The manufacturer, you for not paying attention to what your machine was doing, your pharmacy because it relayed some information and somebody wasn't paying attention. There's a lot of problems right now with what they're trying to do, but still we're right in the early stages. The ethical implications of this are gigantic as well. You could track all sorts of information from inside your body in terms of, okay, this person has X condition, they need Y treatment, this is going to cost Z amount. There's a whole cascade of effects that it can have there. And then at the bottom there, I put the fear of the gray goo, but we're not there yet. Nobody's talking about combining these with nanobots yet, but I thought I'd put it in there just to let you guys know. And where are we now? Implantable microchips were designed first in the 1990s for a way of monitoring insulin levels in conjunction with an insulin pump. Implantable drug pellets were designed in the 1970s to varying amounts of success, depending, same thing on fatty tissue and stuff like that. The rates of application of the drug are different. And this is kind of tying the end of it together, so if you have any general questions after this, or if you have questions just about that last section, we'll get them after this. Moore's law says that every 2 years or so, technology doubles, their computing speed doubles. So technology is advancing at such a rate that the cost of technology is going down. Because you have to think of it in terms of a computer. This is the way I think of it. If you have a computer that's designed this year, people want the newest, they want the fastest, they want the best. That means the computer from last year, the price goes down. To apply that to what we just talked about here, what we have today is nanotubes, nanotechnology, a variety of different technologies. What are we going to have tomorrow that's going to drive the price of this down? Driving the price down will increase innovation, research dollars, everything. And just to tie it all together, advancements in medical technology and drug delivery will serve to improve the quality of life for people who are suffering from a variety of conditions. Whether or not they take any of these methods that we talked about today and run with them is different, but one way or another, it's going to serve to make us all healthier. And I figured somebody was going to ask me this at some point, so I just thought I'd put it in. Do I think that humans will eradicate disease? I pulled this from the Center for Responsible Nanotech. I'll give you guys a second to read it over. All right. So diseases are often opportunistic. If we eradicate one, something's going to move in and bring up that niche. Like I said before, my background is in cell biology, biotech, and I learned a lot about evolution along the way as well. So if you guys have never heard about the Red Queen in either context, that's what we're talking about here. The Red Queen was in Alice in Wonderland. You could read the quote there. She runs faster and faster just to stay in the same place. The Red Queen hypothesis says host-parasite relationships are the same way. They're constantly evolving, we're constantly evolving, but we're staying in the same place. And that's the end.
So, more questions? So, do you believe in a deterministic world? Because that then means really that we shouldn't try to do anything, right? Because it's all been determined what's going to happen anyway. And some of these quotes kind of support that, right? I mean, that if you think it's futile to cure one disease because it'll just be replaced by Well, the next, don't you think there's some generalizable solutions where you may not just learn how to cure one disease, but a whole class of them?
Definitely, yeah. Absolutely, sure. And if we just give up, if you buy into the Red Queen like I do, we're going to stop running and they're just going to catch up and destroy us anyway. So to me, you have to keep going, you have to keep running, you have to keep looking for answers to all these questions, or else.
In order to add, also to add to that, it's not just about curing the diseases, because you don't have to only just cure them. We can also look for ways to manage them, to live with them easier.
Definitely,
so it doesn't necessarily have to open a new niche by eradicating it, like how we almost did with polio, for instance.
Yeah, sure.
Okay, other questions? So, all right, I think we should go on now to Nicholas Lee. So yeah.
There's one.
Yes, maybe the